PhD on the Bushveld at Rhodes University in 1992. He taught igneous petrology and economic geology at the universities of PE, at that stage Pretoria, Chukotimi, Western Australia, and Oulu in Finland. He's professor for ore geology at Cardiff University in the UK. His research deals with petrological, geochemical process, and mafic, ultramafic igneous systems that contribute to our understanding of continental magmatism, mantle evolution, plate tectonics, and the formation of magmatic ore deposits, including PGE, nickel, copper, chrome, and VTM deposits. And with that, I would like to hand over to Prof W Maier, who will be talking to us this morning on the emplacement and crystallization of Bushveld magma, that is new geochronological and microtextural data. Thank you.
Thank you, Sifiso. I guess I have to share my screen now, right?
Yes, please. Thank you.
Can you all see me? My screen, I mean?
Perfect.
Good.
Thank you.
Excellent. Yeah, so welcome everybody. Good morning. Wonderful spring here in Cardiff for the last 2 months during lockdown. So it's been not very difficult to live through that. I'm gonna present some new geochronological and microtextural data today to you. The geochronological data is my student Kate Abenithi's work, which she performed at the British Geological Survey with Simon Tapster in Nottingham. And she's working in industry now, so doesn't have time to present these data herself. I will then also add some microtextural data that we've been working on for some time now with a number of collaborators, Sarah Barnes in Chicoutimi, Steve Barnes in Perth, and Dani and Dave Reed. In 1986, Cox and Singer published a bulletin with the Geological Survey, United States Geological Survey, in which they argued that the origin of the Bushveld ore deposits has been largely resolved. Now, most of us who work on the Bushveld complex, I'm sure we would agree that this view has been very optimistic, and that there are lots of issues with most of our geological petrological models that that don't fit yet, that need to be worked out. And this is a very simplistic model of the classical view by which the Bushveld PGE reefs and chrome and vanadium layers also may have formed. So a rain of sulphide melt droplets triggered perhaps by magma mixing or fractionation sank through a relatively quiescent magma chamber to the top of the cumulative pile. There's been a number of permutations of this model and variations, frequent replenishment of the chamber, and various other variations. But I think most researchers today would agree that this is overly simplistic view, and particularly in the last decade or two, there have been a number of new ideas put forward to explain the reefs. Now, in 2016, Jim Mangel from Toronto published a paper in Nature Comms that started a really vigorous debate. They produced geochronological evidence that some of the Bushveld layers, notably the UG1 and the MG4, most clearly, are really out of sequence sills that were emplaced after the under and overlying layers. Now, they were not the first, of course, to propose that some of the layers intruded as sills, but they were the first to back this up with geochronological data. which many geologists feel constitute more solid evidence than perhaps you know, major and trace element geochemistry. They used chemical abrasion, IDTIMS, geochronology. This is the really the standard these days, and other geochronological studies on the Bushveld complex recently by people like Scotes and Friedman or Amensee in Tübingen have used the same method, and so have we. So there are major methodological advances in geochronology that have been going on there, such as the chemical abrasion that leaches zircons and removes lead loss, the use of the earth time tracer to correct for the lead mass fractionation during the ionization of the material. Now, and I have to get rid of, sorry, I have to get rid of my own view here because I can't see it. The model and the interpretations of Mangel et al. Of course, have been very controversial, and there have been a number of authors who criticized those or questioned those. And on the right, you see a plot here published by Rice-Latipov in 2017 in the South African Journal of Geology, summarizing the age data from Mangel and also Amensee for the upper critical zone. The key critiques have been that the data resolution is quite coarse, often coarser than the actual timescale. So in other words, the some of the zircon ages within individual samples do not overlap. Mangel's work, Mangel's study included multi-grain analysis. In other words, for some samples, they put several zircon grains together and then dissolved them. So that, of course, might mix different temporal signals, which do exist, as I will show you in a moment. The lower main zone weighted mean ages of Zee and Mangel are different by almost 1 million years. So that's a bit of an enigma why that would be so. Is there sample bias? In other words, how many grains really need to be analyzed to make sure that that you cover the entire real spread in ages? And then the argument is that the out-of-sequence layering that Mangel suggested is really only clearly resolved if the averages are considered. So these may, of course, contain unresolved heterogeneity, as we will show in a second. So what needs to be done now, we decided, that was a major part of Kate's PhD thesis, is that the reproducibility of these results needs to be tested. Can we find out-of-sequence ages elsewhere, for example, and particularly in northern limb? That's where Kate was working. Do the zircon populations of single units capture simple or complex thermal histories? And I will show you in a moment that there's lots of evidence that they're very complex. And what can we say about the timing of the flood reef relative to the eastern and western limbs? This is very important for exploration. In particular, we want to, as Dani will show you a little bit later, the flood reef and in fact the entire flood reef, although it's been correlated with the upper critical zone since the days of Morensky and Wagner, these sequences are in some regards very much different from the normal upper critical zone, notably in terms of the much higher sulfide and PG budget. So this is what Kate then set out to test in her PhD thesis, which, by the way, is openly accessible and available through Cardiff University. And if you can't find it, then send me or Kate an email, and we will send it to you via Dropbox or perhaps FastFile. Quite a big document, of course. Here are the ages that Kate, the layers, the horizons that Kate analyzed within the model of the flood reef, and normal upper critical zone that was published in Dani Robler's paper last year. And So yeah, Kate selected samples from the lower main zone, the bastard reef, or the equivalent, what we interpret to be the stratigraphic equivalent of the bastard reef and the Morensky reef, the UG2 chromatite, and then the lower zone 2, and then here a horizon or a unit that we call the PNZ, the pyroxenite norite zone, which might be an equivalent to the lower critical zone or the lower portions of the upper critical zone. We found zircons and good ages, reliable ages in all these units, except for the lower zone. We found no zircons in the lower zone, could not find any zircons in the lower zone sample that we analyzed. So we got to keep trying there. This is a summary of Kate's results, and we're not attempt here to explain this in a great lot of detail or understand it. Even I'm not a geochronologist, but I will try to give you the essence of this. So these are all single crystal analysis, no composites. All of these are single crystal analysis. The median precision is 170,000 years on individual analysis, and the mean. The weighted mean precision is 56,000 years, except for the PNZ, where we have a much larger spread of dates. The greater analytical resolution that the CA-IDTIMS method provides, as you can see, exposes quite a large amount of heterogeneity in individual zircon dates from samples, notably here within the Morensky reef, and also within some of the other samples, PNZ units, the PNZ and the bastard reef. This sort of heterogeneity, of course, might otherwise be lost if the error bars are larger, or if multigrain fractions are used, as has been done in the Mangel study. I go into a little bit more explanations what's happening here. So the data allows or permits multiple interpretations. So this is non-unique. Our interpretations are non-unique. We need to really demonstrate that these are reproducible by producing and looking at similar profiles elsewhere in the Bushveld, eastern lobe, western lobe. That's in the northern part of the northern lobe. A reasonable interpretation in our view is that that the oldest reproducible populations of 3 or more grains, 3 or more zircon analysis, capture in situ crystallization of the magma during cooling. And this is important, I think, these results echo out of sequence pattern that was the out of sequence pattern that was proposed and demonstrated by Mangel. So this is the first very important result of Kate's work, is that out of sequence ages can be reproduced in the northern lobe too. So in other words, the Morensky unit here is sort of sandwiched between the younger UG2 and bastard reefs. We can also look, if we worried about the interpretations that we made there, we can look at the distributions of the data within probability density plots, and this has been done here. And these, of course, are independent of which reproducible population is selected for the weighted mean calculation. And again, these data show us quite clearly there is an out of sequence pattern in the ages. The bastard here is markedly younger than the main zone sample, and the UG2 is clearly younger than the Morensky reef. Also, what's interesting is that the 2 youngest samples, the bastard here and the UG2, in those samples, the distribution of dates can be described by the analytical scatter alone. In the other units here, the main zone, the Morensky and the PNZ, these are the older units. We have greater dispersion of dates. What Kate also did then is she analyzed one sample from the western lobe Morensky reef at Northern Mine, provided by Dave Reed, and to test, of course, whether the blood reef Morensky reef can be is coeval to the western lobe, and yes, indeed, the ages overlap within uncertainty. But the northern Morensky reef shows an even greater, even more extreme dispersion than the Morensky reef of the Flat reef of about 1 million years. So that's a long time. What are the potential reasons for the dispersed ages? There are really quite a number of possibilities here. We feel, and this work is in preparation, and it's also discussed in much detail in Cage's thesis, so I'm not going to go into details here. We feel the most likely explanations for these dispersed states are either protracted crystallization of zircon within a single unit, perhaps by in response to sustained melt migration within the system, or resetting of the uranium-lead system within the crystals, the zircon crystals, during emplacement of later units. So both of these possibilities, of course, require at least localized interaction with later melts within the system. Right. So what does that mean for the model of sill emplacement? What's clear is that sill emplacement does happen. This is an image from Latipov, 2017 Journal Petrology paper. I think it's from one of the Western Bushveld mines, showing the Morensky reef here forming these apophyses, the extensions into its floor rock, sill-like lateral extensions into its floor rock. These can extend for over 100 meters. So these are clearly sills. There's another example here from Andrew Mitchell's paper, is it 2019 or 2020 in the essay Journal of Geology recently, from Wilhelmsbreut, which is just to the west of Union. You can see Morensky reef also forming a sort of smaller scale apophyse into its anorthositic floor. What's interesting here is you can see in brown the staining, that is the sulphides, enhanced amount of sulfides. So it seems that the apophyses contain an enhanced amount of sulfides relative to the normal reef. This is an image from northern mine, Morensky reef. Will Smith, my PhD student, who will talk a little bit later, did some micro XRF mapping of the sample at CSIRO with Steve Pants. And you can see here that the Morensky reef here, then in pink, the chromite, of the basic chromite of the Morensky reef, and the Enorthocyte in blue. And within the Enorthocyte, the footwall Enorthocyte of the Morensky reef, you can see this greenish orthopyroxene-rich layer. That's an apophyse of pyroxenite, of a thin pyroxenite below the Morensky reef that contains, in light green here, enhanced amount of sulfides. So again, just like in the previous example from Andrew Mitchell, the apophyses appear to be enriched in sulfide, and there's less information, not a lot of information on the PGE. I think Rice presented some data in his Journal Petrology paper on PGE in some of these apophyses. They appear to be also often enriched in PGE relative to the normal reef. And I would argue this is the result of hydrodynamic concentration of sulfides during movement of cumulate mushes. This is a picture from the flat reef, from Dani's place. And this is a sulfide-rich, PGE-rich sill in the floor of the flat reef. It's actually a shallow drill core, so this is in the shallow portion of the flat reef, which we would call flat reef then, of course, at Wilhelmsbreut. And so it's a PGE-rich sill in the floor, in the Hornfels floor of the flat reef there, and it runs at 6 grams per ton over 7 meters, so very PGE-rich. You can see there's some variation in textures here, finer grain to coarser grain. So here, again, clearly a sill that has intruded into the Bushveld, a PGE-rich and sulfide-bearing sill. There's another example of a sill. This is an Enorthocyte sill here from Smoky Hills Platinum Mine in the eastern lobe that we studied a couple of years ago. And you can clearly see that it's a sill because it's intruded upwards here into the UG3 chromatite. And we also know that it's a sill that is derived relatively locally, just like the sills, the apophysis of the Morensky reef, because it's connected here to this big Enorthocyte pothole through a thinning out. It thins out, but is connected to the pothole. I'll show you a detail in a sec. Here we go. So there's the margin of this Enorthocyte pothole, and you can see how the Enorthocyte infiltrates and injects here into the sidewall, right above and below the UG3 chromatite, until here it then begins to widen out. So again, a sill of Enorthocyte in this case that originates from has a very proximal origination, rather than, you know, a sill that was in place from a staging chamber. In 2013, what we proposed is that these sills here really form when cumulate slurries, dense cumulate slurries that run on top of the pile of cumulates locally inject downwards into their floor rocks, both chromatites, but also pyroxenites, magnetites, and in fact, Enorthocytes may inject upwards into their overlying slurries or mushes. So the take-home message here would be that the currently available geochronology and the field evidence do not yet provide us with conclusive evidence to constrain the sequence and nature of magma emplacement. So what we need to do, in my mind, is we need to look at a much smaller scale, microtextures. And I'm going to show you the next sort of couple of slides. We'll go into this a little bit. Just check what my time is. Still have some time. So this is data already from my PhD 3 decades ago, where I looked at zonation patterns of cumulate plagioclase. I worked on the UG2 Morensky reef interval, and these are zonation patterns of cumulate plagioclase from 6 random samples across the UG2 Morensky reef interval. And the important thing is that in all, except for this one, in all these grains that I analyzed for zoning, completely randomly, we found clear and very distinct reverse zonation of Enorthocyte content. And these are examples of inclusions, plagioclase inclusions in orthopyroxene from the same interval. Again, pervasively reverse zoned. So this appears to be very common in the upper critical zone. I don't know how common it is in the main zone or the underlying zones. We have done very little work on these, but certainly in the upper critical zone, it appears to be very common. What I also did in my PhD, I looked in more detail then at some of these, what we call mottles in the Bushveld. This is a mottled Enorthocyte from above the UG2 Pyroxenite here. There's a distinct Enorthocyte horizon in the western and eastern lobe. Most of you will be recognizing this. And this mottled Enorthocyte contains commonly these mottles of plano and orthopyroxene, but also locally some olivine in them. And is this evidence for a reactive porous flow? That's what I set out to test. And so what we recently did is we produced element maps. We have a fantastic field emission SEM at Cardiff, with which we can scan here entire thin sections. This is the thin section of that Enorthocyte that you saw a moment ago here. It is in yellow outline, that mottle. And just to briefly explain the colors, in green is olivine, light green and dark green is orthopyroxene. And then in the purples are plagioclase. The dark purple is relatively more albitic plagioclase, and the light purple is anorthitic plagioclase. That's the legend. So anorthitic, what you see here is that this mottle, this mottle has a core of anhedral oikocrystic olivine that is surrounded by a corona, a halo of highly anorthitic plagioclase, cumulus plagioclase, going up to Enorthocyte 95. Whereas the matrix here is typically Enorthocyte 75 to Enorthocyte 80, very typical of the upper critical zone. So this is completely cryptic. When I studied this sample 20, 30 years ago, I did not, of course, see this. We didn't have field emission SEMs in those days. So completely cryptic variation. You can't see it in thin section alone, but it's transgressive if you want. It's a sort of an irregular pod-like zone of clearly non-magmatic high anorthite enrichment in plagioclase. We also did some, when you zoom into some of these grains here, these are the two enlarged portions here, you can see there is a plagioclase. So you see the outline, it's actually a subhedral or euhedral grain even that has here, and in this case, the anorthite-rich portions are brownish, and the in sort of blue-purple is the more albitic plagioclase. So you see the anorthite-rich rim around the plagioclase, all these grains here. And in this grain, you can also clearly see not only does the anorthite-rich zone surround the plagioclase, but it extends into the plagioclase, what looks like cleavage planes. So anorthite-rich zones within the plagioclase grain, and we interpret this to mean that this must be leaching of alkalis by an acidic high-temperature fluid, rather than melting of the plagioclase. We did some with Sarah Barnes and Jacudemy. We did some laser ICP-MS studies of the grains of some of these grains. Actually, we did traverses across the entire model. And the important thing here is that the calcium content of this this grain, this specific grain here, is relatively constant. Look at the enormous zonation in terms of sodium, and also to a lesser degree in potassium. So we're leaching the sodium and the potassium from the margins of these plagioclase grains, leaving the calcium unaffected. The model is also surrounded by coronas of Phlogopite. So the olivine would be in the center here. This is a corona of Phlogopite. There's a corona, a wider and more diffuse corona of amphibole around it, and then also of kleiner pyroxene. The model contains elevated amounts of sulfides, although not a hell of a lot of sulfides, very small amounts of sulfides, but clearly enriched in the model relative to the host rock. It's no quartz. There's no quartz whatsoever in this model. So this came as a bit of a surprise, because if this would be a residual melt pocket, you would expect phases such as quartz, califeldspar, apatite, etc. So no quartz whatsoever. So this to me suggests that the fluids must have been highly silica undersaturated. We did some in situ strontium isotope data here too. Quite a lot of variation in that model in terms of strontium isotopes, suggesting some quite heterogeneous fluid flux going through this, what might be a little fluid conduit. The olivine is also of somewhat unusual composition. It's quite nickel rich for its magnesium, for its phosphide content here. So it has about 72. Phosphide 72, and for this phosphide content, when you compare it to the rest of to global data from other layered intrusions and lavas, then it's very nickel rich. So it appears that if that model and the olivine crystallized in the presence of some fluid, I come to the model in a moment, then this fluid must have been quite nickel rich, could have been quite nickel rich. We find, just to show you that this model is not a freak, we find similar structures elsewhere in the Bushveld. This is from the flood reef here, where we have a little sort of corona structures out there were called by Billy de Clerks 30 years ago, 40 years ago. Here is a zone of Enorthocyte with a little core of looks like olivine here. We have here, this is Billy de Clerks' corona structure, corona horizon below the Morensky reef at Union section in his picture here from his MSc in 1982. So you see the formation here of Enorthocyte with the little cores of olivine in the center. And this is an image here from a Mandelbrot section where we're looking at the pseudo reef overlain by leuconorite and Enorthocyte. And you can see that immediately above the pseudo reef, we have a zone of Enorthocyte, of mottled Enorthocyte. And this mottled Enorthocyte seems to form little plumes or bulges that move, that seem to expand into the hanging wall, suggesting to me that this is a secondary formation here of Enorthocyte emanating somehow from the underlying or triggered or driven somehow, the formation of this Enorthocyte driven by the somehow by the underlying pseudo reef. How I would suggest in a second. Oh yeah, there's the model already, the schematic model. So what we suggest is that the upper critical zone, or certainly this Enorthocyte, was percolated by an acidic fluid that leached very widely sodium and potassium from the margins of plagioclase, producing these reversed zonation. The fluid, of course, was locally dissolved in melt pockets, in interstitial melt pockets within that Enorthocyte. where it lowered the liquidus temperature and caused some resorption of plagioclase. During cooling then, the melt pocket crystallized to form orthopyroxene and olivine. Why would the olivine, we find in some cases the clusters of inclusions of plagioclase surrounded by orthopyroxene, and then of course these sort of assemblages hosted within the olivine, the olivine oikocrystal. So it's possible that, you know, the fluid flux into this melt pocket varied with time. The olivine was, the fact that the olivine is preserved means that ultimately the fluid and the highly volatile rich residual melt must have been expelled, because otherwise the olivine would have reacted back to pyroxene. So that's the model in a nutshell. Other examples, there are other examples of melt flow in the Bushveld. Let me just check my time here. Not much time left. Other examples in the Bushveld. This is the pyroxenite marker in the Eastern Bushveld complex, where you can see an Enorthocyte here forming these little flames and plumes going up through what looks like semi-consolidated or largely consolidated melanorite pyroxenite already. There's the view. It looks like really there's a flame. There's the next one here. So, you know. We have here these Enorthocyte plumes with this images from the flood reef again, and where an Enorthocyte above the bastard reef forms these little plumes moving upwards into pyroxenite. We also in the flood reef find evidence for downward movement of fluids and liquids, and in this case sulphide liquid. And perhaps I skipped this for the moment because it's running out of time here, but there we did some micro XRF work with Steve Barnes, looking at the flood reef pyroxenite, and this is a sample that has about 10 grams per ton PGE over, by the way, over about 40 meters or so, or 30 meters. You can see there are domains here that are sulphide free. Sorry, the sulphides are in light green. Orthopyroxene is orange, and plagioclase is blue, dark blue, and this is clinopyroxene here in purple. So we have domains here where we have pyroxenite with intercumulus plagioclase that are essentially sulphide free. We have other domains where there is lots of disseminated patchy sulphides, but no intercumulus plagioclase. We interpret this to reflect sulphide melt percolation through this layer. So the conclusions then are that we have clearly confirmed that there are out of sequence ages as proposed by Mangel and shown by Mangel. There is a very strong age dispersion in the Morensky reef and some of the other layers. This is something that was not visible in the previous geochronological studies. There is evidence, strong evidence for pervasive fluid and melt flow in the Bushveld upper critical zone. Did this cause this melt flow cause the cumulative crystallization, late stage zircon crystallization, and then dispersion of the ages within the layers? In other words, are these out of sequence ages not related to late stage sills intruded in the sequence, but are they instead related to late stage melts percolating subvertically through the sequence? The fluid flow may have lowered the viscosity of the cumulative meshes, facilitating hydrodynamic sorting and concentration of sulfides in flow dynamic traps. And finally, downward percolation of sulfides on a scale of tens of meters. Sorry for the blending in of an email message. I don't know how to switch this off, so but I'm almost finished. This is the final slide, the preliminary model, and it comes from the paper that has just been accepted in Mineral repository on the flood reef. So we have the emplacement of magma, sulfides formed during fractionation and in situ contamination. The cumulates, these mushes, are then sorted during slumping of slurries along the top of the cumulate pile, and they locally inject into the underlying semi-consolidated mushy cumulates, forming these sub-horizontal apophyses, but locally also very thick trough-like concentrations of sulfide-bearing pyroxenite. That's it. Thank you. Any questions?
Thank you very much, Wolfgang. That was really interesting, and the corona of textures as well. I'll now hand over to the floor for any questions. Just a reminder, please use the raise hand feature on the participants list or at the bottom of your screen. and then we'll take a few questions for the next 5 minutes. Any questions? Okay, Chris, please unmute yourself and ask your question.
Hi, Wolf. Excellent talk. Just that Pyroxenite marker contact. I mean, I think your evidence for fluid flow is excellent, but in the case of the Pyroxenite marker, that looks more like a Rayleigh-Taylor instability with, you know, with the plagioclase just rising up. So I don't think that's necessarily an example of fluid flow. But I think, you know, the rest of your talk is excellent evidence for fluid flow.
When I talk about fluids here, of course, that includes melts, and you know, the Enorthocyte flames, they cross what looks like solidified pyroxenite or melanorite to me, rather than melt layers, Chris.
Yeah. Yeah, no, yeah, that looks more like movement of the actual magma itself rather than the fluid. Yeah.
Hi, Wolf. Steve here. Steve Barnes. Question about your nice talk, by the way. Good stuff. And great to see those nice Maier mapper images. So look fantastic. Your strontium isotope story is interesting. So it looks like you're seeing quite a bit of short-range variability in strontium isotopes associated with the process that's making your olivine-bearing mottles. But elsewhere, there are quite a few papers out now that talk about strontium isotope heterogeneities and are using it as evidence that you've got mixed crystal cargoes and mixing of crystals and so on. What's the sort of the scale of your variation compared to those?
Yeah.
And is it possible that variation is due to fluid interactions as well?
So the scale of variation is very similar on this thin section scale. Is very similar to the scale of variation we observe across the upper critical zone within individual samples, as shown by Yang and others in 2013. So it's a very similar scale, but we see it here within a centimeter. Sorry, what was the second part of the question?
So what's the sort of the range of variability compared to, like, say, for example, that study that you just mentioned, which have been interpreted as, you know, mixed crystal populations?
It is similar to you know, virtually the entire UG2 Morensky reef interval. So there's a lot of variation within a scale of 1 centimeter, matching that what we see over tens of meters in the upper critical zone. But there was a second part of your question. What was it?
No, I think that was it.
Oh, okay. Oh yeah, sorry. You asked something about fluids. Where could the fluids be derived based on the strontium isotope? So that's an interesting question. Initially, I thought that the strontium isotope data means that the fluids cannot be derived from the floor, from the sedimentary floor. You know, instead must be derived from the underlying cumulative rocks. But when you actually look at the data from strontium isotope data from the floor rocks, notably the dolomites, then they happen to have actually surprisingly low initial strontium isotope ratios, overlapping those of the upper critical zone. So what that means is that you know, we cannot be sure that there is not also certainly a dolomitic component in those fluids moving through the cumulative sequence. I think this is an area that needs much more work characterized. I may be ignorant there. There may be work actually that the sedimentologists have done on strontium isotope on the dolomites and the shales, much more extensive than I'm aware of. Certainly aware of it.
Just to comment also, if I may, you were talking about the sulphides in the apophyses of the Morensky reef and PGE contents and so on. I was a collaborator on one of those projects, and what turned up in that was that the PGE tenors of the sulphides was actually highest on the walls of those overturned potholes, and in the sort of the underhangs, you still had sulphide. You didn't particularly have more sulphide, but you still had sulphide. but the PGE10 is in the underhangs was I think up to about an order of magnitude lower than it was on the walls. So there is a difference, but there's still, I mean, there's only a tiny amount of data. It's only been looked at in 1 or 2 places. So there's a lot more work to be done to understand those things.
Thanks, Steve. Related to that, there's a question on the chat. Your impression about the circa 2.06 billion year age established by Hall 2017 from the upper critical zone. That's in the PhD thesis.
Sorry, can you repeat that, please?
It's on the chat. What's your impression about the circa 2.06 billion year age established from the upper critical zone by Hall in 2017 in the PhD thesis?
I'm not aware of the whole thesis. Sorry. And also, I can't see my checkbox. Where is it?
Okay, click that chat right at the bottom there. All right, perhaps you can answer that on the chat as well once you find it. I see Hugo has got a question. Hugo, yes, please go ahead.
Yes, excellent talk, Wolf. Very interesting to see what you have done about the ages there. But my question is mostly about, it's mostly a clarification for me about the fluid. Because there are 2 things that have a conflict. Is you mentioned in one of the answers that when you refer to a fluid, you're talking about a melt, but also you're saying that this melt is acidic. So I'm just trying to comprehend what you mean by an acidic melt.
No, what I mean is that fluids that percolate through the cumulative pile include melts and volatile rich melts and also volatiles alone. So locally, these volatiles, of course, they enter into melt pockets. The volatiles are very acidic, but they locally enter into melt pockets where they lower the liquidus. That's what I mean.
Okay. So if I understand you correctly, then it's probably a fluid saturated melt that is going through.
Yeah.
Okay.
Well, there is fluid saturated melt that's percolating the cumulates, but also volatiles without melt, pure volatile phase, which we suspect is acidic because it leaches the sodium and the potassium.
And the other aspect is when you mentioned that you have leaching of sodium and potassium on the edges of the plioclase, but when you have the transformation from the albite side of the plioclase to the anorthite side of the plioclase, one also needs to have an exchange between the silicon and the aluminum, right? Because the stoichiometry doesn't match unless you move them. So I wonder if the clinopyroxene, for example, could be a repository of aluminum. So if you're moving from albite to anorthite, you need to have some aluminum going in the structure somehow. So I wonder, and you may not have done these, but I expect that some variation in the aluminum content may be seen in the pyroxenes.
Yes,
it may be hard to see.
Yes, no, you see that quite clearly, actually. So basically, initially what we thought is that these pockets simply are residual melt pockets that show late-stage reaction. We see this in cumulate rocks quite commonly, that at the contact between plagioclase and olivine or pyroxene, we form, for example, the Phlogopite, could be explained by that. But when you look actually at the melt pocket, there is a very substantial mass balance problem, notably of things like sodium. Sodium has just gone, all right? It's just taken away. So there's a lot of mass flux in the system. It's not just a residual melt pocket that forms some local reaction. Also, when you look at the pyroxenes, they are very unusually aluminium rich compared to normal upper critical zone pyroxenes. They have up to 2% aluminium. We interpret this to result from in these melt pockets, the resorption of plagioclase by the volatile rich melt, and then crystallization of pyroxene, orthopyroxene, from a relatively aluminium rich melt.
Excellent, thank you.
Right, George, question.
Thank you, Safisa. Thank you, Wolfgang. Just a quick one. is there any direct evidence in the Bushveld for the volatiles, manifestations of the volatiles that you have been talking about?
Yeah, so that's a very good question. And of course, there has been a debate raging now for almost decades, I would say, about this. Alan Boudreau and Ed Matei have been amongst the first to propose that, Bushveld cumulates have been recrystallized in the presence and triggered by volatiles. In those days, this process was not called reactive porous flow, it was called constitutional zone refining or metasomatism. And this sort of model has been criticized for decades now, for example, by my friend Steve Barnes, very vigorously. And one of the main arguments has been that there are just not enough volatiles. There is not enough mica evidence for hydrous phases, micas and amphiboles. And so one of the things that this study here, this detailed study of this model shows is that there is actually quite substantial mica Phlogopite surrounding these features. And one possibility, of course, is that we generally underestimate the amount of hydrous phases in the Bushveld because they can be very, very fine-grained. That's one possibility. The other possibility, and that's something that Alan Boudreau has often argued, is that volatiles, of course, are very mobile, and during crystallization, the last little bits of highly volatile-rich residual melts may be very efficiently expelled from the crystallizing cumulates, so that the evidence that triggered the recrystallization may in the end be lost. So that's the best I can say at the moment. We may be underestimating the amount of hydrous phases in the Bushveld, and there are also some interpretative possibilities to get around that problem.
Thank you.
All right, thanks. Thank you very much, Wolfgang. You may stop sharing your screen now. And yeah, any other questions, as we said, please put them also in the group chat. You can also reply to send privately to Wolfgang or any of the other speakers or any of the participants for that matter. I'd just like to again emphasize, as per the first slide earlier on, just some housekeeping. Please rename yourself with your surname and then your name, just so we can tick your name against the attendance list, and so that we are sure that you are not a Zoom bomber, otherwise you might be kicked out. So please do that. And then, so you'll see that under the participants, where you can rename yourself. And in order to send your ID, participant ID, there's an icon right on the top left that you can click to view this, and then you can send that